All right, so welcome back. So where we left off talking about the action potential, and we talked about the different phases and whatnot. So depolarization and repolarization and hyperpolarization, and we discussed sodium channels and refractory periods and whatnot. And so you may be asking yourself, how are we able to differentiate between the strength of different stimuli, right? And so how do we know that one type of stimulus is actually larger than another? How are we able to perceive these things as being different? So how can we walk into a room and we can smell one thing, right? Um, and then walk into another room and smell the same scent, but it's much stronger, right? So how are we able to distinguish and differentiate between these two uh, stimulus intensities, right? And so it turns out that the strength of a stimulus, so the stimulus strength, causes a different number of action potentials, right? And so the number of action potentials created during a given time is directly related to the strength of that particular stimulus, right? So whether it's a mechanical stimulus or it's a chemical stimulus, it's going to influence the number of action potentials that are generated, right? So if we don't reach threshold, then we will never get an action potential, so you won't see any action potentials, right? If we barely go super threshold, so we're just above threshold, just by a slight margin, then you get some number of action potentials, and the further you increase the stimulus strength above threshold, the more action potentials you get for a given period of time, right? So we're able to distinguish and tell the difference between, the different intensities of some type of mechanical force or sound or some type of smell or chemical stimulus based on the number of action potentials that that particular stimulus generates, right? In future lectures, we'll talk about how some of our receptors that sense stimulus are able to respond very quickly, so they adapt very rapidly. So this is what's known as a phasic receptor, and then we'll also talk about some that adapt very slowly that are called tonic receptors. All right. Now, there's an important clinical correlate to our action potentials, and this is a poison, a toxin that comes from the Japanese pufferfish known as tetrodotoxin, right? And so what tetrodotoxin does is it's a toxin that's found in the Japanese pufferfish. So this is the pufferfish here, and it is a very strong and very potent blocker an inactivator of sodium channels. All right. So if you look at the graph to the left here, this is a normal action potential, right? So postsynaptic response to presynaptic excitatory signaling in the absence of TTX, in the absence of tetrodotoxin. So you have your resting potential, you get a nice stimulus, you get up to threshold, you have an action potential, depolarization, repolarization, hyperpolarization, and then you go back to normal, all right? So what tetrodotoxin does is it binds and inactivates voltage-gated sodium channels, all right? So depending on the amount of tetrodotoxin present, that you ingested from eating, improperly cooked pufferfish, you have a certain number of voltage-gated sodium channels that are blocked, right? So if you block these, you decrease the ability of the neuron to reach threshold, because whenever a stimulus arrives and the voltage changes, it's not going to open the voltage-gated sodium channels because they're being inactivated and they're being blocked by the presence of tetrodotoxin. So what you get is you get paralysis, and by blocking the sodium channels from opening, you block sodium from entering, so you block reaching threshold, you block depolarization, and if you don't have those, then you never get your action potential, right? And so this does cause the death of a few people each year from eating improperly cooked pufferfish. And the issue comes when it inactivates the sodium channels associated with the nerves going to your respiratory muscles. You're unable to take in a breath. You're unable to inhale. All right. So note to self: try not to eat any pufferfish. All right, or at least improperly cooked pufferfish. All right. And so all of this has to do the depolarization and repolarization. Our main goal, if you want to call it a goal, is to ultimately stimulate effectors, right? And so we want to stimulate muscle, or we want to stimulate glands, or something like that, right? And so to do that, you have to make sure the action potential is moving in such a way that it is flowing towards whatever our effector is. It's flowing toward the muscle. It's flowing toward the gland. All right. And so we start at our axon hillock right here. We have a high concentration of sodium channels, and we begin flowing down this axon in this particular way, right? So from 1 to 2 to 3. So we go from 1 to 2 to 3. And so what you have happen is at each point you have depolarization, you have repolarization, right? So you have potassium coming in, and then after it leaves, potassium is flowing out. And that prevents the flow of the action potential from going in the opposite direction, all right? So this area of the membrane is still in a refractory period, right? And so this is preventing the sodium, or it's preventing the action potential from flowing back in the opposite direction. And so instead we move in just one direction way down this way, right? So we move from this area to this number 2 in the middle, we move to number 3, and we continue flowing in one particular direction, right? And by the time the membrane has reestablished its membrane potential, the action potential has already left and is flowing down toward the synaptic terminal, toward whatever particular effector tissue that neuron is going to, all right? So we can have different factors that influence this propagation or this movement of the action potential, right? One of them has to do with the neuron itself, so how wide is it, how thick is it? And the other has to do with a substance known as myelin, all right? So the wider the axon, the faster the propagation, and whether or not it has myelin. So if it has myelin, it's going to propagate the action potential more quickly, all right? And so what you have happen is, if you think of the width of a neuron in the same context of as the width of a highway, all right? If you have a wider highway, you're able to send traffic, and traffic is able to flow faster than on highways that are only 2 lanes, right? Or you can think of it as your merging lane. So if you go from a 2-lane highway and then you merge onto a 5-lane highway, or you go from a 5-lane highway and you go to a 2-lane, the rate of the traffic flow, the speed, is going to vary depending on the diameter, how wide that particular highway is. And so the same thing happens here. Depending on the width of a particular neuron, you're going to get a different rate of flow of the action potential flowing down the neuron, all right? Now, you can also have the propagation of the action potential influenced by something known as the myelin sheath, all right? And so if you look at this here, these two neurons, you have this neuron here on the left, you have this one here on the right. This one is myelinated, this one is unmyelinated. Myelin is a lipid fatty substance that wraps around the neuron, and you see that it does not wrap around the neuron the entire length, but it leaves these little spaces known as nodes, right? Nodes of Ranvier. Okay, so this is pronounced Ranvier. At each node of Ranvier, you have sodium and potassium channels, right? However, where the myelin is wrapped around the axon, you don't, right? So what this means is that the process of sodium channel activation, opening, sodium flowing through, inactivation, and then closing, and this whole process of depolarization and repolarization and using sodium-potassium pump to pump ions on either side of the membrane. In an unmyelinated axon, that has to happen at every single point down the neuron. So it has to happen here and here and here and here and here and here and all the way down this axon, the whole entire length at every single point. So you can imagine that takes time. That takes a long time. It also takes a lot of energy as well, lots of ATP. So if you only have to do that at certain points on the neuron, you're able to speed up the rate at which you can send an action potential. Because instead of it happening at every point along the neuron, it only has to happen at every node. So sodium activation and inactivation only has to occur here. And then it only has to occur here. And depolarization and repolarization only have to occur here and here, and so on and so forth. And so the speed at which and the rate at which you can send an action potential is much faster in a myelinated axon than in an unmyelinated axon, right? And so there are conditions that are demyelinating diseases. A good example is one called multiple sclerosis, or MS. And this is a demyelinating disease that takes away the myelin, and this slows down the impulse conduction of action potentials flowing in the nervous system, All right. Now, this myelin sheath can be created by two different types of cells. One is called a Schwann cell. The other is called an oligodendrocyte that we'll talk about in future lectures. Schwann cells myelinate neurons in the peripheral nervous system, whereas oligodendrocytes do so in the central nervous system. And this is known as, whenever you have myelin, the impulse conduction, it is known as saltatory conduction, right? The Latin term saltare means to jump or to leap. So you can think of the action potential as jumping or leaping from one node to the next, whereas in a unmyelinated axon, this is known as continuous conduction. It has to happen at every single point, and so it takes much longer, okay? All right, so what we'll end with here is looking at two different types of synapses, and we'll come back to these synapses in chapter 9 as well when we talk about skeletal muscles. So you have two types. You have one called a chemical, and you have one called an electrical synapse. These are named according to the means by which the action potential crosses from one cell to the next. If it crosses via a chemical synapse, you have chemicals, right? So specifically, we're using neurotransmitters, right? So we're using acetylcholine. You have these vesicles with the neurotransmitter fused with the membrane. They release the neurotransmitter. It binds to a ligand-gated receptor. It opens, and then you have sodium rush in, right? So this is chemical. This is what we'll look at in chapter 9 whenever we do our skeletal muscle contraction lecture, right? You also have electrical, right? And so electrical, instead of using chemicals, you're using the flow of ions to flow from one cell directly into the next, all right? And so the way you have ions flow directly from one cell to the next is you use a gap junction, right? And so a gap junction is an open, you should recall this from chapter 2, is a transmembrane protein made up of smaller individual units that can open and close, and when they open, they allow sodium and whatnot to flow from one cell to the next very, very rapidly, right? So this allows for a very rapid movement of ions, and so it allows a rapid conduction of the action potential, all right? So you avoid the delays that you have with chemical synapses because you have to have release of neurotransmitter and you have to have binding and opening and the movement, so it's much slower with chemical, all right? So you see these electrical synapses in our cardiac cells, and you'll talk about it in AP2 whenever you do cardiac action potential, which is a different type of action potential than the one we're talking about. And the reason you want this in heart muscle is you want the heart to contract as one functional unit, right? You want the heart to contract as one functional, what's called a functional syncytium, one functional unit. And with a electrical synapse, you get that. You get the rapid movement of ions from one cell to the next through gap junctions that are located in intercalated discs that join two adjacent cardiac myocytes together. In chemical synapses, that transmission of an action potential from one cell to the next takes a lot longer, and so that is not an ideal type of synapse to have in a heart cell in which you're wanting the heart to contract as one functional unit and pump blood very efficiently. You don't get that with a chemical synapse, and so instead you use an electrical synapse, right? Okay, so that finishes our excitable cell lecture. If you have any questions about the material, please reach out. Please shoot me an email, and I will be happy to answer any of your questions, and I will see you in the next video.